Our presenter this evening, Dr. AlexSweger, is an assistant professor of biology at Hartwick College. He specializes in behavioral ecology and arachnology. His work investigates issues in sensory ecology and behavior using primarily wolf spiders as model organisms. We're looking forward to learning more about this unique topic. Welcome, Alex.
Thank you, Becky. And thank you to the Delaware-Otsego Audubon Society for inviting me. And for everybody, thank you to all of you who showed up. I always like a chance to talk about the work that I do. So I guess I should get my screen shared here. I think that should be okay. Looks like we're good there. All right. So as Becky said, and thank you again for that great introduction. I study behavior, primarily in wolf spiders, and I just recently joined the faculty at Hartwick College. So most of the work I'm talking about tonight is actually stuff that I've done previously in my career, but it should hopefully communicate some ideas that I have about the kinds of topics I'm looking forward to pursuing in the coming years with students from Hartwick. And along those lines, I want to start by, before it gets lost in the shuffle, at the end of everything. And also because hopefully I won't have to cut myself off, but I want to say it now, some acknowledgements of some of the folks I've worked with over the last few years. Two previous mentors. So a lot of this work was done alongside or under the supervision of both Dr. George Utz and Dr. Matthew Persons, who are in these two pictures right here. There's Matt, there's George. And a whole slew of fantastic undergrads. The five here are the ones that were primarily involved in some of the work that I'm talking about today. And I'll mention them as I get to those particular projects, and all the institutions I've worked with and funding sources. There's a lot that goes into making sure this work is able to be done. And the work that I do primarily is in, as Becky mentioned, behavioral ecology. So I like to start typically, I know there's a lot of people with a lot of different backgrounds in the audience or that attend these programs. So I wanted to at least establish what that is. Behavioral ecology is ultimately about how behavior evolves and how the environment's connected to that. The reason that it's interesting to study behavior, I think, beyond just having a general interest in watching what animals do, is that behavior is extraordinarily variable. It's arguably the most variable aspect of an animal's biology, an organism's biology, but particularly within animals. And it can be variable even within a species, within an individual over its lifetime. That variability is the very thing that makes it interesting from an evolutionary biology perspective, because there's a couple things that come along with that variability. Number one, it responds very quickly to selection pressures. So the same forces that drive evolution, behavior is like the first thing that changes usually before color and structure or the genetics or developmental features, behavior changes. And that means that you can really quickly get a nuanced view of what forces are acting on the evolution in that species. Number two, lots of different factors in the environment affect behavior, and you can measure differences. You can sort of look at how an organism interacts with its environment by looking at its behavior and what changes in that behavior as the environment changes. And that's the ecology side of this, the interaction with the environment. And then ultimately, because it's so variable and so nuanced, if there are changes to the environment, you can almost always see them, even if they're very nuanced, first in changes in behavior. And that can be anything from pollution, toxins in the environment, to gradually changing climate. All these things that might subtly influence an ecosystem and environment, one of the first things we're going to see is changes to behavior. So I think it's really, really useful feature to look at in biology. And beyond that, I'm particularly interested in communication. Most of what you're seeing there on the right is some sort of behavior associated with communication. And the sensory systems that those animals use and their communication modes adapt to changes in the environment as well. So I typically look at changes as they influence communication. The second question I always have to answer is, why spiders? And there's a couple of reasons for that. One, from a sort of practical perspective, it's cheap work. There's less paperwork. It's easy to access. There's large populations in every major terrestrial habitat on Earth, basically. Certainly in all the areas where most humans live. They're invertebrates, so while we treat them ethically and humanely, there isn't as much federal paperwork to gain access to using them, and it's easy for students to work with them. It allows for a lot of larger studies, for quicker, smaller studies, so it's very adaptable to things that are also variable, like student interest. And not the least, not the least important thing is that people are also always curious about them. Sometimes they're grotesque, so they're grossed out. Sometimes they're really interested, but they always are curious. On the right, there are two public consumption articles that were written about some of my work. I did not give them those headlines, but that just gives you an idea of the kind of sensationalism that comes out of doing this work. The real reason that I like working with spiders is in terms of the biology and evolutionary biology of the species, they're abundant and they're diverse invertebrates. So they're located in, as I said before, almost every major terrestrial ecosystem. Within those ecosystems, they are active and efficient predators. They're responsible for something like 20 to 40% of biomass removal, depending on the species or depending on the ecosystem that we're talking about. They remove a ton of insect pests because they are predators and because they're such efficient predators. They are also food sources for larger organisms. Almost every food web that you're looking at in a forest, a field, spiders are somewhere there in the middle. They're consuming some of the lower invertebrates and they're being consumed by higher vertebrates. So that's a really important place in a food web. Beyond that, they're really interesting models for sexual selection. Some of the evolutionary forces that lead to things like peacock's tails and all kinds of weird communication modes. Spiders are really interesting. They have super complex genitalia. They have super complex behaviors. They're very, very weird. There's been a lot of strange things that have happened in their evolutionary history that has made them really interesting when you're looking at how they communicate, how males and females communicate. And not the least of, there's a lot of things that are interesting, and not the least of which is the fact that there's also the potential in a lot of species for sexual cannibalism. Females sometimes eat males. Sometimes the percentage is very low. Sometimes it's as high as 80 to 90%. And that's a really interesting force for a biologist because it's kind of this crossroads between survival and reproduction, which are two really strong forces in evolutionary biology. Any male that wants to pass on his genes has to go up against and basically convince a potential predator. And that means that there's a really, really strong selection pressure on efficient communication. That male has to say very quickly, I am your species, I am male, I am mature, and I am a good partner. And that means that the communication modes get honed very, very quickly. And that's what I'm interested in. And all this stuff varies across spiders as a whole because they're very diverse. The group that I'm particularly interested in is wolf spiders and sort of how they stack up against other groups. There's around like 2,400 species worldwide. There's about 200 in North America alone. Most of them are relatively small, not more than a couple millimeters. There's a couple species that get up to about the size of your palm. They're all pretty much harmless in terms of venom. They're non-web building, they're ground dwelling. They have good vision. They're dichromats, kind of like your dog, so they can see in color, but it's muted colors. They're very active hunters and they're good parents. They carry around, the females will carry around an egg sac and then also carry around spiderlings that you'll see on her back until they disperse. So there's a lot of interesting features of their biology. Some of the things that I've looked at and the things projects I'm going to talk about tonight are using 3 different species of wolf spiders and investigating 3 different topics, but they all kind of relate back to those things I said at the beginning about how we use behavior to answer bigger questions about the evolution of signaling and communication and changes to our environment and sort of how those things are interrelated. So the first one I want to talk about involves a species that I worked on in my graduate work. This species here on the right. Basically what happened is I went to University of Cincinnati, worked with George Utz, who was studying a very well-studied species that he had studied for 35 years at that point. We went to go collect them for a typical research season, and we came across this species. It's a little larger, about a couple inches across, an inch or two across, really. And we didn't know what it was. I, being the diligent graduate student, used some keys to try and figure out what species we were dealing with here. And it keyed out to this species, Galactosa gulosa. So then I went digging to see what we knew about it. And this is all I found, which there's probably people with different amounts of familiarity in what the scientific literature looks like, but this is not much, suffice to say. And almost all of this is just anecdotal. It's, as you can see there, it's field notes from the early 1900s, a couple of descriptions of one or two individuals that people found, almost nothing experimental had ever been done. So of course I was intrigued. And what made it more interesting is that despite these being just notes, they all mention one particular thing, that they make that they're audible. They purr. There's this weird soft purring sound you hear, and that it's loud, supposedly. You can hear it at 10 feet or more, or up to 6 meters, which is almost 20 feet. I mean, this would be a very loud sound for a spider to be making. So I was completely enamored of this idea as a grad student, and set out to start building a dissertation around studying this species. Starting with getting our own recordings of this species, because there was nothing extant from these early papers in the early 1900s that actually gave us a recording of this species. At the time, we now have, where present day, we have much more complex techniques for recording these things. I won't go through all the sort of nuances of the details there, but we used a couple different recording devices. We did video recording of these individuals. We used a microphone above the arena to record if there was any audible sound. And we used what's called a laser Doppler vibrometer, which is kind of just a really expensive laser pointer that basically works as a microphone for vibration. So it's directly recording the sounds being made by that spider. So we get a recording of their vibrations that they're making in the paper that they're on, and any airborne sound that we're hearing as a result. And we can stimulate most spiders, male spiders in particular, to create these sounds by just giving them some paper that a female has walked on. There's a lot of chemical cues that they leave behind. So we basically gave males some paper that had been sort of cued with female chemical compounds, and then recorded what he did. And I'm going to show you a recording. So hopefully, we just tested this earlier, so it should work. You'll see a video of a male courting like he believes there's a female nearby. And the sound you're hearing is actually coming from this laser Doppler vibrometer. So this is the vibrations that are inside the paper that he's creating. And Becky and Susan, feel free to let me know if that sound isn't coming through. I can always go back and we can try to tweak it, or I can post these separately and go from there. You probably noticed two behaviors there, two distinct things it was doing. That little purring, chirping sound it was making, that's actually made by a structure called a stridulatory organ. We call it stridulation. It's more or less the same thing that crickets do, but they're doing it with their wings. And it's done at the base of this little appendage called a pedipalp, right near the mouth parts. There's two of these. And at the terminal joint, the final joint between these last two segments of that limb, there's a little file and scraper mechanism. So on one side is like a ridge structure, and the other is like a pick. It's almost like taking a pick down a washboard or a comb. And so they'll just oscillate that joint back and forth, and they'll get that chirping sound. We've known about this organ since the 70s, so this isn't shocking. The other thing you saw was percussion, which in this case is they take their abdomen and they repeatedly drum it against the substrate. So everything we've seen up to this point was very cool, I think, but not shocking or revelatory. This is pretty standard courtship for a wolf spider, but it is loud. And so what I'll do is I'll play you the same clip, but this time the audio you're hearing is from the microphone in the room. And it's going to be soft, it's amplified, but again, consider the fact that this is a spider, not something that you typically associate with airborne sound. So we're hearing sound. And you can hear it in the room. It's soft, but it's louder than you would expect for a spider. And I can say, I can't confirm those anecdotes saying you could hear it at 20 feet away. But George and I both went out in the field, and at night, on a clear night, you stand still in the quiet, you can hear them around you at about a 3 to 6 foot radius. Soft, but it's louder than you would expect for a spider. And even weirder than that, for those that may have some sense of arthropod anatomy, spiders don't have ears. So that raises another question that's interesting. Lots of animals have ears. Certainly all vertebrates have some sort of a tympanic membrane like we have inside our ear canal. And even things like insects, sometimes it's on a leg, sometimes it's on a part of the body, the thorax or regions of the abdomen, but they have membranes that respond to changes in air pressure, sound. Spiders have nothing like that. Not that's ever been found. So beyond just the sort of weirdness of a spider making noise is the obvious evolutionary question, for me at least, why would they make a sound that they can't hear? Especially if it's a sound that they're supposed to be making for females. Like I said, spiders have no, as far as we know, organ associated with hearing airborne sound. They do, however, have extraordinarily sensitive vibratory organs. So all these different organs here are an anatomical representation from Frederick Barth, showing all these different organs that are associated with sensing vibrations. And obviously the vibration was quite strong. And this means that webs are an extension like an eardrum, and these things that are on leaves are using vibrations to communicate. And there's arguably some behavioral evidence that they're so sensitive that as long as they're on something that vibrates, they can hear a representation of their environment, not through the same means that we do, but they do sense it. At least it seems that way. So there's a lot more to follow up on this in terms of what I was interested in as a graduate student. In any event, we characterized the rest of this signal, published it, did some of the quantitative measurements, and alongside sort of looking at these different elements, we also simultaneously compared the sort of vibratory signal and airborne signal. So that sound you heard in the paper and the sound you heard in air. And they correlate. I don't have the data up here. I have lots of extra slides if anybody wants to see any of that data. And it's in this paper. But they correlate. The louder the vibration, the louder the sound. They happen at the same time, and they're directly correlated in terms of amplitude, which is not shocking. That's just a principle of physics. If the source is coming from a vibration, the louder that vibration is, the louder the sound that results will be. But the question still remains, at least for us, why is the vibration so loud? Because it's louder than it should be for a spider of that size. It's another study that I did looking at sort of relative amplitudes across different sized species, and these guys are disproportionately loud for their size. Because it's a courtship signal, the obvious next step would be to look at what's actually happening between males and females in this species. And so with the help of a student, Corey Vaughn, who made it sort of, this chapter of my thesis was sort of a big project for him. We worked together to design a set of experiments where we paired males and females together, did the same set of recordings, and then looked at how that differences in these courtship behaviors related to mating success to see if we could get an answer to this question. So I'm going to play you another video. This is the last one. There's a male and a female that are separated. They can see each other and they can hear each other through the substrate, but they can't get in contact yet. They will later in the trial be able to access each other. You'll hear the male through the LDV courting, and you'll see the female in the background responding. And we've labeled some of the behaviors that we tracked over the course of this experiment to sort of give you an example of some of the things she does to signal to that male. So you saw a couple things there. And without going into the nuances of this big data set that we generated, we looked at all these different aspects of what the male's doing, how often he's doing stridulation, how often he's doing percussion, the relative amplitude of each. And we looked at all those different behaviors you saw, quantified them, created some definitions for what we were calling this and that. And then took that big data set and just said, what's important when it comes to whether or not these two end up mating. And the short answer is, the louder the male is, the more likely he is to mate. That's one of the biggest pieces that came out of the male side of it. The female side, a lot of those behaviors showed up a lot, but that waving and tapping she does with her front limbs, the more she does that, the more likely she is to allow the male to mate. And more importantly, they're correlated. This data set's messy, but it is significant. The louder the male is, the louder his courtship is, particularly that stridulation, which is what you hear audibly, the more she taps back, and the more likely they are to end up mating. Which suggests to me there is pressure on the female side of things for males to get louder. As they get louder, they're more likely to get mates. And as a byproduct, they're also more likely to produce airborne sound. That might give us an answer to why the sound got so loud. It doesn't yet answer the question of whether or not a female can actually hear it. And the problem, the implicit problem, is as long as they're on paper, there's going to be both vibration and airborne sound. So we have to isolate that if we want to get an answer. So the next step then, in another experiment, was to basically design a little arena where we put females down on granite, and then from a randomized side, left or right, we would play them sound, isolated from the granite. And we could use the LDV to make sure we weren't transmitting any vibrations while we were playing back this sound. And so we would present these sounds to females. We would give her either a blank, so no sound, just ambient noise in the room, white noise, so a sound that was about the same amplitude or volume of the male courtship signal, but it wasn't relevant, it was just noise, or the male signal. And surprisingly enough, there was no response. Females don't respond when they're on this granite. However, if you put paper down in this arena, you get a response. This on the left is how rapidly females began movement. So they start moving around the arena. Females are usually pretty stationary most of the time. They're not actively wandering around, but they start moving around. A higher proportion does, and more of them overall begin to move and move more quickly. They spend more time moving, and over here, they spend more time in association. They move close to that stimulus. And the reason for this, ultimately, is if we take the LDV and go back, even if we're not generating a vibratory signal directly into that paper, when you play the sound, airborne sound, you get something of a reproduction of it in the actual paper. So what you're seeing here is there's a lot of noise in the lower frequencies, but there's nothing. Here's our white noise, and here's the actual spider signal. And this isn't being played directly into the paper. We can do that, but we didn't. We just played it from a speaker nearby, but it creates a representation of that. So it just reinforces this idea that they are able to hear their environment as long as it translates into a vibration. Now, we haven't yet answered the question. I've got lots more experiments. My dissertation obviously included more than just these that try to dissect out what might be happening. But ultimately, I'd like to think that this might be giving us the early signs of a system where we can try to investigate how a mode of communication evolved. There's a lot of evidence in other species that vibration was a precursor to airborne sound. We know birds and lots of other insects use airborne sound, but the earliest emergences of airborne sound came from a vibratory source. And this might give us some clues. This species might give us some clues about how that first happened and what the selection pressures might have been to lead to the emergence of a airborne sound from a vibration. But is a long way away to actually come up with solid evidence of that. This is just sort of the beginnings of it. I'm going to change gears here to sort of talk about a different type of question. While I was still working with George, this time as a postdoc, so yay, I finished my PhD, still supervising some other projects in the lab. We changed gears over several years because of an undergraduate question. So this project, the effects of environmental moisture on courtship signals, was focused on a different species, a smaller one, only about yay big, about the size of maybe a quarter. Is called the brush-legged wolf spider, Schizocosa ocreata. It is a spider that George worked on for more than 40 years. He just retired this year, so a silent round of applause for him, finishing up his 40-plus year career at the University of Cincinnati. It is a very abundant ground-dwelling wolf spider. extremely high densities in forest habitats. And you may notice it also has something weird going on here. The males actually have, beyond just producing vibrations, the same way Gladycosa did, they also have these dark, hairy forelimbs, and they wave them around at females. So they have both a visual cue and a vibratory cue. And those signals are similar to what we saw in Gladycosa. The patterning is a little different, but they do both stridulation and percussion, and they wave these foreleg tufts around. And what's more important, previous studies in George's lab have shown that they're flexible in what they use. So they can wave these legs around, they can make vibrations, but they shift the proportion of those depending on what substrate they're on. Shearer Gordon, a previous student of George's, showed that if they're on something that vibrates, like a leaf, they'll do a lot of vibrations and a lot of leg waving. If they're on something like rock or soil that doesn't transmit those vibrations, they do more of the visual, more of the waving. So a lot of plasticity, flexibility in this species. That's all very cool. Talking through this with a group of undergrads, just trying to sort of propose some different projects, an undergrad very reasonably said, this might be a dumb question, which is something undergrads always say, because they always think their question is dumb. They said, what if the leaves are wet? To which George said, however many years they've been working on this species, they'd never really thought about, what about keep the leaves, but add moisture? And that's not an irrelevant question, because at the time where we were working in Cincinnati and throughout the range of this species, that area, the Midwest through the Northeast, gets something between 50 and 60 inches of rainfall annually. It varies obviously pretty heavily, but that's not an insignificant amount of rain. And specifically in Cincinnati, we saw peak rainfall in the season right at the times in late May to early June when this species was breeding. So when these communication signals would be the most relevant. And there's a lot of evolutionary implications to this, because this species is under what we might consider a scramble system, where females have basically in the population reached maturity, have already reached maturity as males are beginning to mature. So they're already there and ready to go as the males are just hitting sexual maturity and ready to court. And females will typically only mate once. So males are immediately out there trying to compete for access to females. And if the rain that's happening at the same time is affecting their communication, it makes sense that males would need to be able to overcome any sort of obstacles that get in the way of their communication. So what we set out to do was essentially answer those two questions. Number one, does it affect their vibration? So what we did was we basically took the spider out of the equation, used a vibratory source, a little piezoelectric element, which is similar to what makes your phone vibrate. And we could play back male signals through that into a wet leaf and record it with our LDV at different distances to see if there's some drop off in a dry leaf versus a wet leaf. And then we took it to the other side of things. We did just the spiders. We put males and females together in wet or dry environments, little mesocosms or small containers. And we just looked at what the courtship looked like and whether or not they successfully mated. And what we found is there is an influence, and they have an ability to deal with it. There is a drop off, a significant difference in the transmission and the volume of their courtship, their vibration, over a set distance when you're dealing with a wet leaf. It mutes the signal, and it creates a lot of extra noise in that signal. The leaf is heavier. It doesn't transmit those vibrations as well. So then that question seems pretty obvious. Then the answer to that seems pretty obvious. The next question then is, does this translate into differences in mating success and what males are doing to communicate to females? Turns out it very much does. On the left here is how quickly males began courting, and you'll notice not much of a difference. Pretty much within the first minute, all but 75 to 100% of the males were courting, regardless of whether the leaves were wet or dry. But on the right here, you'll notice that the number of those visual displays, waving that visual ornament around, is significantly higher when they're on wet leaves. So just like when they're on granite or something else that doesn't transmit these signals, the vibratory signals very well, they can shift to more of a visual signal, the visual component of how they communicate. And this translates to recovery of some of their mating success. There was a significant difference. Males are still more likely to convince a female if they're on a dry substrate. But something like 40 to 50% of the population was still able to successfully secure a mate, even if the leaves were wet. And this makes sense too. I mean, I didn't talk about the data here, but females also prefer what are called multimodal signals. So females prefer both. They like to see and hear the male rather than just one or the other. But for males that are dealing with an obstacle, an impedance to their vibratory communication, the visual signals, if they can shift to them, can recover some of that success. So on top of one species and one project giving us some insights into the evolution of these signals, this is a nice example of how we can use behavior to, again, try to evaluate how variations in the environment have shifted male signals. And it could also, arguably, and this is an argument that George has made for a long time, provide some evidence of why both signals evolved in the first place, why you would even want to deal with a visual signal when you're already making sound. There are advantages to having both in some of these species. Now I'm going to heavily shift gears. This last piece, this was after I had gone back to my alma mater, Susquehanna University, and worked with Matt Persons again and some great undergrads. And we took this sort of behavioral evidence to its sort of logical conclusion in terms of environmental change by looking at what's happening in an agricultural system and what's happening specifically with chemical herbicides on a species that's living in this. But we're still focused on how behavior can tell us something about it. And to sort of set the scene for this project, this is a major issue, as I'm sure I probably don't have to explain to a crowd that's attending a program like this, but we use a lot of pesticides. And pesticides is a generic term for a chemical that's used to eliminate any unwanted species. You can subdivide it by what it targets. Insecticides target insects and other arthropods. Herbicides target plants. Fungicides, fungi. There's things like acaricides or acaricides that target mites and nematicides that target nematodes. Suffice to say, though, herbicides are a huge component of that. Almost half of almost half of the pesticides that we spray annually on fields are herbicides designed to kill unwanted plants, weeds. And that keeps growing. It has grown over the last 50, 60 years, and it continues to grow. This is data from about the 60s to 2012. And in that 50 or so year period, the amount of herbicides in terms of millions of pounds has gone up tenfold. And it continues to go up. And there's some side effects to this. One of the reasons it continues to go up, but also a side effect, is that it's driving the evolution of weedy plant species. And we see this in insects as well. We see pesticide-resistant species emerge. And you can see this nowadays if you go around any sort of agricultural field that's been sprayed. It's dead for a while, but on the edges, you always see a few species popping up. And so the sort of traditional solution is just to crank up that dosage, to spray more and to spray more often. Other downside of this is that there's effects on non-target organisms. There's a lot of, every major company that produces herbicides attests to the fact that it doesn't have effects on non-target organisms, but the studies are usually done in-house and the measurements are pretty rough at best, if that makes sense. So there isn't a nuanced view most of the time of what the potential effects could be of these chemicals being sprayed on non-target organisms. And if we're focused on herbicides, animals are non-target, period, because they're not supposed to affect animals, they're for plants. And there's been a lot of evidence that there are herbicides that have been affecting non-target animal species for a while now. Multiple different pesticides were implicated as part of the sort of complex cause of things like colony collapse. Tyrone Hayes from Berkeley has a long history of studying atrazine and what its runoff from agricultural fields has done to the sort of gametic tissue in frogs. It has ultimately chemically castrated them at very small chemical levels. There's a lot of diverse effects. And in terms of the species that are near and dear to my heart, as I mentioned at the beginning, and it's certainly true in agricultural systems, spiders are major predators in agricultural systems. Radioactive tracing studies finds an accumulation of those tracers in spiders in agricultural systems. They're estimated to remove as much as 40% of insect biomass from those systems. They are abundant. And one species in particular is a wolf spider species that is massively abundant. Arguably the most abundant ground spider in agricultural fields in the eastern United States. This is Pardosa milvina. You can find it around here. You can find it all across most of the United States. It's only about yay big, so it's relatively small, but you can go out there and after a cornfield has been plowed, if you walk around through there and look closely, you will see dozens of them scattering with every footstep. You know, a rough anecdotal estimate by Matt and some students put their density at something like 16 adults per square meter, which would put us in the tens of thousands per acre in these agricultural systems. And so they're likely consuming something on the order of 2/3 of a million insects per week per acre of land. They're a massive part of this ecosystem and a big part of biological control. And more importantly, they're out there year round in these systems. So they're definitely going to be out there during any number of spraying events. And so we set out to sort of answer questions about what might be happening and if there's a nuanced way to look at it. Because if these herbicides that are being sprayed don't outright kill these species, are they doing something else that isn't detectable unless we look at something nuanced like behavior? And so we set out to do this using a great piece of property owned by Susquehanna University that they've dubbed the Center for Environmental Education and Research. The university proper's over here in the top right. But right over here is this large portion of land that's also owned by the university where there are some sites. There's some buildings here that are used for freshwater ecology research. There are some plots here that are used for teaching first-year ecology students. There's a solar array that was put in recently to help make the campus more green and reduce carbon emissions. But right next to this center, inside this white box, is an old agricultural field, a former soybean field that for the last 10 to 15 years has not been touched. It's been allowed to sort of return. Actually, it's not. Sorry, I have that wrong. It's been farmed with alfalfa, but it has not been sprayed. It was basically turned over to try and measure some things like no-tilling practices and other ways to sort of deal with biological control without using pesticides. And so there is no spraying history on this field for the last 10 to 15 years or so. And then conveniently enough, shortly or just across the street, there's another plot here that is a more traditional farming plot, also owned by the university, but it is allowed to be farmed by a local farmer who worked with the university and was, number one, very willing to work with me and Matt, and also kept meticulous notes. So he had a record of exactly what he planted and what herbicides he had sprayed on that field for the last 10 years at the time. And there were 5 major herbicides that were sprayed on this field. Sometimes nothing was sprayed. Other times a single one was sprayed. Other times all 5 were sprayed together. So this gave us a nice little study system. We could take individuals from each of these 2 fields, males and females. We could get commercial copies. All these herbicides are available commercially. Sometimes they require some permits, but we could get access to all of them. We could distill them down to field relevant applications, and we could expose spiders to them. What we would do is basically store spiders in these little souffle cups, like you would store condiments in, and we would calculate out our pre-emergence dosages. So these are lower level dosages designed to be sprayed on a field before planting. Converted it down so that a single spray, or roughly the volumetric equivalent, was sprayed on that container. We added the spider, closed it off, and then proceeded to continue with the experiment. So these spiders were not directly sprayed. The environment they were situated on, the substrate they were standing on, was sprayed. We then monitored these species over a 52-day period. I shouldn't say we, it was 4 absolutely fantastic students that did this. And over that 2-month or so period, they also weekly looked at feeding trials, looking at how quickly males or these spiders captured prey, changes in weight, egg sac production. And once in that 2-month period, they paired some males and females to look at behavioral responses. And obviously, over the course of all this, they were monitoring survival in each of these groups. And when I say each of these groups, this is where the impressiveness of what these students did really comes into sharp relief. If you're playing the home game and tracking this, for those that are somewhat savvy in experimental design, we had 7 treatment groups because we had 5 individual herbicides. We had a distilled water control, and then we had a mix where we put all 5 herbicides together. We had spiders from 2 different sites, those that had previously been exposed to pesticides and those that had not, from the 2 different field sites. And we had males and females from each of those 2 sites. So that's 2 times 2 times 7 for 28 independent treatment groups. And our target sample size was something like 50 by the time we were done. We landed somewhere in the low 40s for most of these treatment groups. We're talking about 12 to 1,500 spiders that were monitored every day and every week for 2 months. And to give you an idea of what that looks like, okay, even in little souffle cups, we basically had to take over an entire lab. These 4 students were putting in 20 hours at least on this every week for 6 weeks of their summer. This data set wouldn't be possible without that kind of an effort. So this is a nice little, alongside the sort of ecological implications of this data, it's a nice example of the kind of efforts that young undergrad students can really, what their efforts can do to contribute to the sort of our broader scientific knowledge. So I'll show you some of this data. This is a very big, very complicated data set. Here's the lethal data. So what we're seeing here is the proportion surviving from 100 down to 0, basically, over this 52-day period. And just to sort of give you a frame of reference and why there's a little bit of bias in what companies say is lethal, this is where most acute toxicological studies stop. This is where they stop measuring, about a week to 10 days. Here's what our study showed over the course of about 2 months. Up here, there is no difference in these herbicides. Down here, however, stuff shakes out a little different. The rim sulfuron treatment was not fundamentally different from our distilled water control. 3 of them, atrazine, glyphosate, and S-metolachlor, actually did significantly better, which is actually because we believe, hypothetically, because of a process, physiological process we call hormesis, that I can explain if people want an explanation on that. But it's something that's well known and we think might be the cause here. But most dramatically, everything together killed all the spiders within about 3 weeks. And mesotrione by itself killed basically the entire population in 2 weeks. And there were a lot of nuanced behavioral differences that I won't necessarily go into. We saw differences in how quickly they captured prey. We saw differences in egg sac production and weight change. That was a complex mixture of which site they were gathered from, what treatment they were exposed to. But we set out to look for these nuanced behavioral measurements, and instead we came away with some pretty dramatic lethal effects. And to sort of bring that home, the mesotrione is manufactured and sold by Syngenta to the tune of about $400 million a year. It goes by the sort of commercial names of Callisto and Tenacity. We went back and triple checked our calculations. This is the kind of data set that when you see it, you think we have to have made a mistake in our dosages. And as far as we can tell, and it's been confirmed independently multiple times, we did not. We were using the field relevant dosages. It's mostly used on corn, mesotrione. We dump about 4.5 million pounds on it. That was in 2018. I don't know if there's more recent data than that, but it's probably gone up if historical trends are any indication. And these compounds have to be reapproved every year. So whether or not this data contributes to that, I don't know, but we're trying to put it out there and demonstrate that there are effects to these compounds that are not immediately clear and maybe subvert or belie the data that's been provided about their relative safety. To sort of bring this back to the bigger picture of all this, it's just one more way that we can use behavior. Assume that maybe we didn't kill off everything, we can look at more nuanced changes in behavior to say there's still a difference in this exposure. This change in environmental toxicity may have effects on things that are still equally important to the survival and evolutionary success of this species. How it hunts, how it reproduces, how it when it becomes sexually mature, which could also be another thing that's impacted. So the sort of take-home message, I guess, here is the reason I'm a behavioral ecologist is because there is a diverse set of questions you can answer about the evolution of a species or the evolution of animals in general in their communication, in how they interact with their environment. And you can get at that in a very nuanced way by looking at behavior. And I'm looking forward to continuing projects like this as I begin my career at Harvard. Thank you for attending. And if you have any questions, I'll gladly answer them with what time we have. I also put my email up there, so feel free to contact me with additional questions. Thank you.
Alex, I do have a question that was asked. I have a few for you. This person wonders how the first species of spider would react to wet substrate. Do they have a visual cue ability?
Yeah, that's a very good question. And it's one that we considered. It didn't make its way into the study system that we were looking at. But I don't know. There isn't much of a visual cue. I mean, there's some visuals that they do as they're moving around. But I don't know that they would have much to fall back on. Perhaps the increase in the amplitude could overcome that. I haven't directly tested whether or not that louder amplitude successfully overcomes the noise, but I don't have a good answer for that. It's a very, very good question. They're living in the similar habitat as Schizocosa, so it's a valid question. I don't entirely have a good answer for that. It's a great question.
Thank you. Also, do you know if spiders get louder in the rain or during different seasons?
That's a good question as well. If they get louder in the rain. I mean, measuring, yeah, I mean, there's some complicating factors there. I don't know. Obviously, with something like Schizocosa, they shift towards more visual, but the relative amplitude of their signal is a tough thing to measure, because to do that, you'd have to measure on a wet leaf, which is obscuring the signal to begin with. We use things like signal to noise ratio and different measurements acoustically to get at that. But you would almost need to know like a physiological effort that they're putting in to get at a question like that, because I don't know that you could tease it out of just the sound that you're recording there. But it's, again, a valid question. And I suppose we could get at that by sort of simulating louder or softer signals in a wet or dry leaf. But whether or not that translates to what they're doing is unclear. We could only get really at physically what that would then look like. But it's not something we pursued. Again, these things spiral out of control pretty quickly. We're trying to just narrow in on a couple of key features, but it's definitely a question that could be pursued in the future.
Oh, thank you. Also, what were they feeding the spiders during the study?
Yes, typically we feed species in the lab. We feed them house crickets. They're regularly available. We can sort of control their diet. We bring the crickets into the lab. We usually feed them a mix of something like total and dog food, the crickets, to make sure. And then they're fed to the spiders on a weekly basis. You can supplement that diet with a couple of different things, but generally they do, after they're a certain size, they do fine just on crickets, and they continue their behavior, as far as we can tell, similar to what they do in the field. They're probably eating some more diverse diets in the field. But in terms of maintaining a lab supply, we're feeding them crickets typically. It makes for a nice controlled species to give them.
Interesting. How did you catch so many spiders?
Well, I mean, in some species, Pardosa in particular, we collect during the day. As I said, you can literally go out there. Typically, we'll collect after cornfields have been mowed or before they've been planted, with the permission of local farmers. They usually don't care if we're going out there collecting spiders and not interfering with the crops. Once those fields have been plowed and you've got sort of an X cornfield or X soybean field, every step, I mean, it's just a matter of grabbing them off the ground. We use little plastic vials, scoop them up. For the larger species, we do what's called headlamping, where we go out at night. This might creep some people out, but we go out at night, and at the back of their eyes, these big posterior median eyes, they have a reflective membrane called a tapetum, or tapetum lucidum. It's the same thing that like deer and cows and things like that have. Cats have. Cows, I don't know. Cows definitely do, actually. Most animals that are out active at night, it's a light gathering membrane to increase visibility, brightness, but it reflects. So anybody who spotted deer with a spotlight, you can do the same thing with a spider. You put a little headlamp on, and you'll see little shimmers in the leaves, and you can spot. I mean, this is going to sound crazy, but you can spot a tiny spider like that from 20 or 30 yards out. It's impressive. Yeah, students are always a little blown away when we take them out for the first time and show them how we're going to collect, and they don't realize how effective it is to do that. But yeah, that's one of the ways for some of these species that are not as dense, they're still large populations, but they're not, hundreds of them every square meter. We use a headlamping technique. It depends on the species, but yeah.
Thank you. Mm-hmm. How far? I just had the question here. Hold the phone. How far do the spiders travel? Could they have moved between fields?
Yes, I think possibly. That's certainly something. This. I'm guessing one of the reviewers from that paper is in the audience. That's a very good question, and one that reviewers brought up, and we honestly can't say for sure that they didn't. They're not insects, so they're not winged. They're not, and as adults, they're probably not ballooning. So it seems unlikely that they were traveling those distances. And there is a highway that goes, not a highway, but a major road that goes between the fields. So because of the changes in habitat, we kind of assumed that, but we did kind of hand wave and say, we don't know for sure that they didn't move. And there haven't been a lot of studies that have accurately tracked the exact distance that an individual wolf spider will travel in any given amount of time. We tend to assume that if they're in this local habitat, like a field, where they have food and they have shelter, they're probably not actively moving, hundreds of yards away. But that may be happening. We don't know.
And one more question. It sounds like the wolf spiders are outsourcing something like hearing to other substances that can act as membranes, like externalized eardrums. Are there other species that employ that method of perceiving sound dash vibration?
Yeah, there are quite a few. I mean, there are species that utilize substrate is massively important when you're dealing with any amount of sound. So, I mean, this could be anything from, it can be both for sound reception and production. If anybody's familiar with what a mole cricket is, we don't really have them this far north, but down in like Oklahoma, there are these big, thick crickets that chirp, but they'll make a burrow that kind of looks like a tuba horn in the ground, stick their butt in the hole, and then create a sound that travels out and amplifies that signal. And there's lots of other animals that, even if they're not creating something as like a satellite dish or sound, they're careful about what substrates they're on. Even things like elephants, which in the last decade or so we've begun to understand how much they use vibration on top of being acoustically oriented. And you'll see elephants change the positioning of their fat pads on their feet when they think they're detecting the rumbles of another elephant. So there's lots of animals that do this. My hypothesis with these species is that it's the leaves. They're traveling around on leaf litter, and that leaf litter readily vibrates, both in terms of receiving sound and also producing that sound. There's a data set with Gladicosa that I didn't present here tonight, but when you change what substrate they're courting on, you lose both the vibration and the airborne sound. It's only when you're on something like a leaf that you actually get that sound. Some animals have that built in, others do not. Crickets have specialized membranes so that when they stridulate, you actually hear it quite loudly. Spiders do not, but the leaves could be playing a part in creating that sound.
We have time for one more question, and then I think other questions can be directed to your website, as you mentioned. One more question. Would you please give a little more info on why some of the pesticides had a lower lethal impact than distilled water?
Yeah, we don't know. I've dug into that a little bit. It depends on what they're targeting in the actual plant. I want to say mesotrione targets, I think it interfaces with tyrosine. I want to say, please don't quote me on this, but I believe, and that can if it messes with the sort of enzymatic cascades involved in tyrosine, that can mess with molting ability in these species. and failure to molt successfully can be lethal. But I don't know that we necessarily saw a huge dramatic change in their molt failures where they just died. Again, that is one of the limitations of being a behaviorist or behavioral ecologist is I don't know the toxicology and physiology enough to say in any amount of detail what it is that's killing. But all we can say is that with under control conditions, it did. And it was a dramatic effect compared to every other group. In fact, you notice that it was actually significantly worse with just mesotrione than we mixed them all together. So that compound, for whatever reason, is aggressively lethal for that species. And Matt, I know, has continued to study this in other species and has found that it's not as dramatic in other groups of organisms. So other species of spider or guilds of spider do not get impacted the same way that wolf spider did. Even things like earthworms that are down in the soil and absorb things through the body wall didn't respond. So there's something in the physiology that it's reacting with, but I don't know enough about the chemistry involved. I don't know that any of us do to say for sure. It's certainly important that we get at that mechanism in some way, but I don't.
Let's sneak one more question.
Sure, of course.
Did you tease out any behavioral changes in the spiders that lived after the treatment or anatomical changes?
Yeah, I mean, I can fire through a couple of slides here, but I won't go too nuts here. I mean, there were, I'll give you some of the quick hits here. This is some of this other data, and it's very complicated very quickly. Some of the herbicides caused some weight gain, some caused weight loss. There were sex-based differences in weight gain and weight loss, depending on the species, or depending on the herbicide that was used. So it's not just males and females were equally affected by the herbicide, but in some herbicides, females and males gained or lost weight differently. It's hard to say what's going on there. There were a bunch of complex interactions with how quickly they captured prey, but the biggest overall trend was we saw that spiders from different fields were different in their ability to capture prey, which we thought was odd. Teasing out exactly what was going on is kind of complicated. And once you get down to the sample sizes we were left over with, it's hard to make any real connections to what is actually happening here. The last one I'll say is the females from these conventionally sprayed fields produce significantly fewer egg sacs, which might be relevant. So the one that was actually sprayed regularly versus the one that had not been previously exposed to herbicides, we saw a difference in egg sac deposition. It's tough to say if that's actually irrelevant. All we can say is that we saw a significant effect in this study. There's also, for those that are more savvy with ecological studies, this is technically a sample size of 1. It's pseudo-replicated to a certain extent because you would need to do this across a series of fields that had not been sprayed and a series of fields that had to really get at if that was the key cause. So it's complicated. I will, if anybody specifically wants more info on that, I can go back here. The paper here is Ward et al. In 2023. It's lethal and sublethal effects of common herbicides on the wolf spider Protosa bovina and ecotoxicology. We go into a lot of detail about trying to evaluate that massive data set. That's about as much as I can probably provide here, but if people want more, I'm happy to continue to try to answer questions about that paper. But there were a lot of complicated effects. For those that survived, we saw a lot of differences, weight change, and primarily in weight change and ability to capture prey.
There are unanswered questions that will come your way.
Sounds good.
Alex, I want to sincerely thank you for opening our minds to this fascinating topic.
Well, I'm always happy to share about my research. And again, I'm happy to answer questions. So feel free to shoot me an email. Thank you.
I want to go outside and listen. Do I have a prayer of finding them? I'm an oral person. Can I hear them?
Possibly. It's got to be quiet. They come out early in the spring, which is the other thing that was sort of built into my hypothesis. They're coming out earlier than most other species. Mm-hmm. A lot of spiders will overwinter as subadults. They'll hit a certain threshold of growth. In winter will come, they'll go down and sort of go dormant. And then in the spring, they'll come out, they'll eat a lot, they'll reach maturity, they'll mate. This species reaches maturity in the fall and doesn't mate. And then it stays mature over the winter. And so it comes out early in the spring and mates almost immediately. So it's a little different phenologically than a lot of other species. I couldn't tell you. I will say, if you hear them around here, please tell me, because I have not. These species are very this species is very localized, and I have not found a good population of them at high densities since I was in Cincinnati. I know they're in all those early papers are from the New England area, so I'm certain they're around. I just have to find them, and I would love to work with them again. So I would definitely appreciate it if you found them.
To quote one of your viewers, a fascinating topic and a fascinating subject and a terrific presentation. Thank you.
Well, thank you. I very much appreciate it.
We appreciate you. Take care.
Thank you.
Bye-bye.